So, hello and welcome to Bio 212, Anatomy and Physiology 2 at Norwalk Community College. And we're in Chapter 20, which is Cardiac Structure and Function. And we're moving on to cardiac muscle tissue and the cardiac conduction system. So, at this point in time, I need to remind you that there are three different types of muscle cells inside of the body. And we're focusing on, obviously, cardiac muscles themselves. And what you want to be doing is you want to figure out how these are different than the other cells. Meaning, if I was to draw, say, a skeletal muscle cell, I would probably draw something that looks pretty linear, right? And it has multiple nuclei, and they're peripheral. And then you have contractile fibers. That's what those lines represent, contractile fibers. Okay? If I'm drawing smooth muscle, I'm drawing something that's slightly angular, spindle-like, if you will, a single centrally located nucleus, right? And then you can't see contractile fibers. Skeletal muscle fibers, up here, linear, slightly angular here. Cardiac muscle fibers are somewhat angular as a result of the way that they are arranged, but they have a single centrally located nucleus. And they have striations that you can see. My cartoon is horrible here. I apologize. Okay. Well, you'll see in the that cartoon in the next slide. Okay. But what's important here are these sort of characteristics. And one thing you want to find at each of these things is these things that are called intercalated discs or intercalated discs, and within the disc we have these things that are called desmosomes, and they're holding everything together. And what's happening is what they're holding in place, orienting properly, are these gap junctions, and those gap junctions are going to allow ions to pass between the cells, which means we don't have to worry about charges entering and exiting the cell. Once we actually cause a charge to move into one cell, it just seamlessly passes through to the adjacent one. These cells also have mitochondria that are larger and more numerous than skeletal muscle. And that should make sense, right? We need to have continuous catabolism of energy sources to make ATP. So the mitochondria are slightly different. However, the contractile arrangements, as I suggested to you, are identical. Let's get rid of the cartoons. Let's move on to the next image here. And this is what's inside your textbook. And here's what I was trying to draw for you, sort of slightly angular cells that will actually have these abutments, and these abutments will form intercalated discs, and these intercalated discs have these gap junctions in them, and these gap junctions allow the ions to pass through. And similar to our skeletal muscle tissue, the sarcolemma, the outside plasma membrane of the cell, has T-tubules that invaginate into the actual cardiac muscle fiber with a single centrally located nucleus here. What I want you to pay attention to is how sparse the sarcoplasmic reticulum is inside of this. And that's significant because if you remember from AMP1, we store calcium inside of here. And that calcium, we need a 2 here, right? Calcium moves out of here in a voltage-gated manner. So that calcium now goes and binds to the troponin and tropomyosin complex here that will allow muscle contractions to occur. So that's actually really important, meaning we have limited availability to calcium. Where are we going here? Inside our heart, okay, I'm going to draw a heart for you here. Change color. Go down here. Yeah. And here's a heart, right? Here is a heart. We have 2 atria, we have 2 ventricles, and I'm going to let you know that there are autorhythmic fibers inside of the heart, the first of which is going to be the SA node, SA, which is sinoatrial node. We have an AV node, atrioventricular node, and it's attached to this thing that's called the bundle of His that travels through the interventricular septum. And coming off of that, we have a series of fibers, and those fibers are called Purkinje fibers. Okay, so we're going to AV. These are His. Bear with me here. Sorry. His. And these are just going to be called Purkinje fibers, PF. What I want you to understand is that the SA node and, to a lesser extent, the AV node are responsible for generating what we call pacemaker function, right? And what happens is it repeatedly generates action potentials that can trigger heart contractions. And you can imagine that if it's essentially deploying an electrical charge here, and you remember that all cardiac myocytes have gap junctions, the arrow that I'm drawing is the representation of the amount of distance the electrical charge will have to occur to cause a concerted atrial contraction to push blood from the right and the left atria into the right and the left ventricles. Okay? While that's happening, that charge finds its way to the AV node here. It then causes a charge to pass through that fibrous skeleton, allowing the bundle of His to transmit information through the interventricular septum. That information is, of course, electrical, which will then be transmitted through the Purkinje fibers, which will then activate concerted muscular contraction regulated through electrical stimulus to push blood from the right right ventricle and left ventricle into either the pulmonary trunk to go to the lungs or the aortic arch to travel to the body. Okay. And in total, though, what you've done is you've created a conduction system. So what does it look like? Here's what it looks like inside of your textbook. Here's a heart that's been reflected, and these are artistic interpretations of the electrical transmission that travels from the SA node all the way over to the edge of the left atrium. It will activate the AV node, which then travels into the atrioventricular septum, so atrioventricular node here, into the interventricular septum here, where you can see the left and the right bundle branches that have been activated by that bundle of His, and then travel around to the left ventricle and the right ventricle. Their picture is much prettier than mine, but you can draw mine quickly. You can't draw theirs quickly. Okay? So this is the idea. Notice that we have to have the SA node depolarized. First, it regulates the depolarization of the AV node, which then regulates the depolarizations of cardiac muscle fibers inside the ventricles. Okay? So what we're going to be doing with this, though, is the SA node is that natural pacemaker. It initiates about 100 depolarizations per minute. That should say minute. That's wrong. Per minute. Okay? And what's going to happen is the regulation of these is through the autonomic nervous system. So this is what establishes the fundamental rhythm, right? Roughly 100 beats per minute. And what's going to happen is either, I'm going to draw this as, I have one coming in here like this, acting on, that is the end bulb of, say, parasympathetic P. And we'll have sympathetic, let's change colors here just for effect's sake, right? And then over here, we're going to have S, parasympathetic, so this is going to release epinephrine, that's epi, norepinephrine, and over here, we're going to release acetylcholine, A, C, H. And that's important because if we want to change how fast or how slow the heart is beating, you have to use these neurotransmitters that act on the SA node that then change everything else inside of the heart. And by that I mean the rate at which blood exits the heart from the ventricles. How it happens, of course, is we actually have to have a series of depolarizations that become action potentials that will then cause contraction. And what I think you should probably understand for the individual cell level, right? Okay, is that you're going to have some depolarization event, a plateau event, and then a repolarization event. And this is for individual contractile fibers. So that's one cell. Okay? And what's going to happen, of course, is here's our depolarization. That is the plateau. And this is going to be our repolarization right here, right? So why does Mother Nature do all of this? Get rid of that. The depolarization utilizes voltage-gated fast sodium channels, which will then allow for the rapid increase in the rate of the charge traveling across the surface of the cell, right? So that's what this is. And notice the way it's being described. That's important. You need to understand that we have to use voltage-gated fast channels, which means it's the electrical event coming from the conduction system that starts this. That plateau period, okay, you have voltage-gated slow calcium channels. Let's go like this, okay, which moves calcium from interstitial fluid into the cytosol. This means from outside of the cell, okay? And then what's going to happen is voltage-gated potassium channels will now begin balancing out the calcium inflow with potassium outflow. And that's what begins the repolarization. So you actually have an electrical neutralization occurring here. Okay? The repolarization is going to be additional voltage-gated potassium channels are going to open, and calcium channels are closing as a result of the change in the charge, okay, at the membrane. Okay, that refractory period is going to last longer than the contraction itself, and it ensures, this is going to sound kind of obvious, it ensures that tetanus maintained contraction cannot occur, right? Because if your heart just beat once and stayed contracted, obviously blood flow would decrease, and that's shown to you here. Not the tetanus piece, but these are all of the steps that we've just worked our way through. And I want you to notice you're starting at roughly minus 90 millivolts. We get to plus 20 voltage-gated fast sodium channels, voltage-gated slow calcium channels, voltage-gated potassium channels, and then all of a sudden we get back to normal. And notice I pointed out to you that calcium channels are closing because look what's actually happening to the voltage gate here. That voltage-gated calcium channel can't open until we actually get to roughly this range here. Okay? So if that's what's happening in an individual cell, this is what actually happens as the sum of all of the electrical events. And this is our electrocardiogram, ECG or EKG. And there are, your textbook says there are 3 recognizable waves. There are 3 recognizable waves for this piece of information that we're looking at. And it's interesting, each time I look at this, the P and the T waves change inside your textbooks. And that's probably good because the sensitivity is increasing is what's happening there. But essentially, in blue over here is where atrial contraction is occurring. And I'm challenging you to put a piece of paper at the end of this and follow that down. And you're going to see that electrical event has to precede contraction event, which means electrical event, if we draw the straight line down through here, electrical event for the ventricles has to occur before ventricular contraction can occur. And that makes sense. What doesn't make sense in the tracing is this, is if this is depolarized, where's our repolarization? Okay, where is it? You can't see it because there's so much electrical activity that's occurring inside the ventricular depolarization at that time that it's lost to that noise. And this is what you're seeing here. This T wave is the repolarization of all of these cells. And so there's such a great deal of electrical activity that, there's such a great deal of electrical activity that the actual inflection point looks like it's a depolarization. And this is the result of trying to get all of the electrical information for the different numbers of cells scaled to fit into an interpretable readout. Okay? There's a craft to reading these things that I do not profess to have mastered at any point in time in my life. I do have a understanding of what it's measuring and how important it is, though. Okay? So all of these things I just spoke about, right? So systole is going to be contraction and diastole is going to be relaxation, or systole and diastole. East Coast, West Coast, Middle America, makes no difference. Systole is contraction, diastole is relaxation. You're going to learn eventually these are going to be lub and dub. and cardiac action potential arises in the SA node, and that's when the P wave begins. Atrial contraction, atrial systole. You have the QRS complex occurs, but notice where it's happening. The action potential enters the AV bundle and it exits out over all the ventricles. That electrical activity, whoops, where did I go here? Darn, I just wanted to show that to you quickly. That electrical activity is what I was just talking about. And that's what masks the repolarization of the atrial output here. Okay, repolarization is that T wave, and then ultimately you actually have ventricular relaxation, and that is diastole. Okay, where are we going to go with this now? Well, your textbook has a picture of what's going on, so let's take a look at that. Here's one. Take a look at the green here. This is the SA node depolarizing, and that's what's actually happening there. Repolarization. I apologize. Okay, where you actually have the contraction here. Now there's a pause as it's traveling through down into the ventricles, where we're then going to initiate ventricular contraction. Okay, wham, that's what's going to happen here. You're going to push blood out into the pulmonary trunk and the aorta. And then all of a sudden, this actually begins to repolarize, and that's the T wave you see there. Then there's a pause, and then you start the entire process over again. If you want verification of this, just simply put your hand on your heart, and you can begin to feel these. Okay? You can sum all of this together by what's actually happening inside the heart, and this is where things get to be a little bit more complex, right? Because now we're talking about electrical activity causing contractile activity, which changes pressure inside of the heart. And I wanted you thinking about pressure before, so this is where we're going to sort of dig in for the next minute or so, maybe two. Regardless if we are in the atria or inside the ventricles, if that muscle contracts, you should increase pressure. When the muscle relaxes, it's going to decrease pressure inside that space. Okay? During the relaxation period, both atria and ventricles are relaxed, meaning at the end of that heart rate, meaning the entire cardiac cycle. Now, obviously, the faster the heart beats, the shorter the relaxation will be, okay? And therefore, both systole and diastole can shorten their lengths slightly. They can't ever be completely stopped, okay? Or made so brief that you can't efficiently push blood from the atria to the ventricles to the systemic part of the body. Okay? Hopefully we'll see how that happens in this next figure. Okay, so this next figure inside your textbook, some days I really like it and some days I really don't like it. And the parts that I like about it, like, let's take a look down here, way down at the bottom. You can see this traces essentially some of the stuff we've seen before, and you're going to notice the vocabulary has changed just a little bit. Atrial contraction causes isovolumetric contraction. So atrial contraction takes an area of high pressure, pushes it into an area of low pressure. The consequences here is you've changed now the pressure inside here, and now you've increased it, and therefore you cause contraction. That contraction leads to ventricular ejection, and that ventricular ejection leads to a drop in pressure here, but notice what's happening inside here. Increasing pressure inside the atria, and then we fill the ventricles again, and then we start the entire process over. What's happening inside the heart? Well, you can trace all of these back. Just simply follow the colors back. Contraction from PQRST pushes the blood into the ventricles. Does that make sense, right? Likewise, if you jumped all the way over here, you actually have your ventricular repolarization occurring here, and that's when the ventricular ejection is occurring, because contraction had to have happened. So you have atrial systole, electrical event, ventricular systole, relaxation period. So it takes about 0.08 of a second to complete one of these entire cycles. And remember, a moment ago I said you can change that relative to the amount of need the heart actually has. So let's just take a look at two more things here. And that's going to be, one, the volume inside the ventricle. So we sort of end, meaning at when it's full. So you actually have atrial systole. How much blood did we push in there? About 125 mils, right? And by the time we finish ventricular systole, notice what we're down to, just about 55 mils, which means that contraction pushed that volume of blood. Let me make sure I highlight that for you. This is the volume of blood that was pushed out of the ventricles. And notice it means there's always still blood inside the ventricles. Well, hopefully you understand what that means already if this is a pump. The last thing I want you to pay attention to is here. If we finished the heart sounds, you actually have the opening and closing of valves. And I want you to sort of push yourself to go in and take a look at what S1 and S2 look like inside your textbooks. And I will ask you about that inside class. Okay, it's a good place to start.